Hello everyone, welcome to the presentation. Today we are going to discuss about hydrophobic interaction chromatography, theory and principles of hydrophobic interaction chromatography. If you are new to this channel, consider subscribing and click on the bell icon to get notified. Hydrophobic interaction chromatography, or the HIC, is a powerful technique for the separation and purification of biomolecules. This technique was initially termed as salting out, and it is widely used for protein purification, for isolating protein complexes, and also for studying protein folding and unfolding. The hydrophobic interaction chromatography utilizes the reversible interaction of protein and the hydrophobic ligand for the separation of protein mixtures. So it is due to the reversible interaction of the hydrophobe and the protein makes it a separation technique, and the proteins will be separated in the order of their increasing surface hydrophobicity. The sample molecules containing hydrophobic and hydrophilic regions are applied to the HIC column in a high salt buffer. So the proteins has amino acids which are having hydrophilic and hydrophobic regions are mixed in a high salt buffer and loaded onto the HIC column. The salt in the buffer reduces the solvation of sample solutes, and as the solvation decreases, hydrophobic regions become exposed and are absorbed by the matrix or the HIC media. The more hydrophobic the molecule, the less salt is needed to promote binding. So this is basically to expose the hydrophobic region in the molecule. So that's the reason salt is used. So if the protein is more hydrophobic, then only less salt is required to promote the binding. Usually a decreasing salt gradient is used to elute the samples from the column in the order of increasing hydrophobicity. By decreasing the salt gradient, protein can be eluted from the column. In certain cases, mild organic modifiers or detergents are also added to the elution buffer to promote the elution of protein from the binding matrix. The hydrophobic interaction chromatography is explained in three different theories. So they are the salting out and hydrophobic interaction, thermodynamic theory, and the surface tension and van der Waals forces. So the explanation for this hydrophobic interaction chromatography is done based on these three theories. So let's see one by one. First is salting out and the hydrophobic interaction theory. The elution or precipitation strength of an ion is described by the Homfmeister Series. Small highly charged ions are strong precipitals, whereas organic acids and bases have more stabilizing effect in presence of proteins in solution. The term chaotropic refers to the ability of an ion to produce chaos in the water structure. The anti-chaotropic salts such as ammonium sulfate and sodium sulfate expose hydrophobic patches on protein by removing highly structured water layer, which usually covers these patches in the solution. As a result, hydrophobic residues on protein molecule can interact with the hydrophobic ligand of the matrix. Salts can also reduce the solubility of proteins by shielding charge groups which normally keep the protein apart in solution. When the electrostatic charge on protein molecules are shielded, these molecules can easily interact, form aggregates, and eventually precipitate. The solubility of different proteins is reduced to different extents by salt addition. So that is the role of salt in promoting the precipitation or the hydrophobic interaction with the matrix. Chaotropic agents disrupt the intermolecular forces between water molecules, allowing proteins and other macromolecules to dissolve more easily. Chaotropic agents interfere with stabilizing intermolecular interactions mediated by non-covalent forces such as the hydrogen bond, van der Waals forces, and hydrophobic effects. Let's see the second theory on hydrophobic interaction chromatography, that is based on the thermodynamics theory. The thermodynamic theory of hydrophobic interaction chromatography directly relates to Gibbs free energy equation. The phenomena of increased interaction in the presence of salting out ions is explained by a higher gain in entropy when water is transferred from the surface of a non-polar molecule to the bulk water. In hydrophobic interaction, the entropy change is of greatest importance. If hydrophobic molecules such as aliphatic carbon chains are immersed in water, the monolayer of water molecules in contact with the carbon chain will be in higher order than those in the bulk of water. If two or more hydrophobic structures come together, the surface that has to be covered by ordered water molecules decreases. Some water molecules join less ordered bulk water and the entire system gains the entropy and thus decreases its free energy. And this state will be favorable for energetic reactions and that will be promoted. So that is the basis of thermodynamic theory on hydrophobic interaction chromatography. A third theory suggests that van der Waals forces are responsible for the hydrophobic interaction. This is supported by the fact that these forces should be increased as the order of water increases in presence of salt. In HIC, media is described based on the ligand density and the available capacity. It is the actual amount of protein that can bind to the media is called as the available capacity. And if flow rate is included in the defined condition, then it is called as the dynamic binding capacity. And the dynamic binding capacity is based on these factors like salt concentration, flow rate, temperature and pH. Here are the some of the examples of dynamic binding capacity of different HIC matrices. The characteristics of a good HIC matrix includes high binding capacity because the larger area will result in high binding capacity. And the media selected should have physical stability and chemical stability, and the matrix should be inert, so it should not interact with the buffers or other reagents. It should be inert and it should be physically stable and chemically stable. The most commonly used salts in hydrophobic interaction chromatography are ammonium sulfate, sodium chloride, potassium chloride and ammonium acetate. The ligands used in the hydrophobic interaction chromatography media has alkyl or aryl groups. The alkyl groups include butyl, octyl, ether, isopropyl, etc. One of the example is butyl S, and for the aryl groups it is phenyl. Example is phenyl 650. The alkyl shows pure hydrophobic character, whereas aryl shows mixed behavior. And the ligand attachment to the matrix in HIC is done through glycidyl ether. Let's see the binding and elution conditions. The binding is done at a high salt concentration, typically 1 to 2 molar ammonium sulfate or in presence of 3 molar NaCl. And the elution is performed by reducing the salt concentration. Elution of proteins from the HIC will be in the increasing order of hydrophobicity. So these are the references you can refer for more information. Thank you.